Professor SNAB, where we make A-level biology as simple as a cell. So today we're understanding the structure and function of polysaccharides such as starch and cellulose, including the role of hydrogen bonds between beta-glucose molecules in the formation of cellulose microfibrils. This is a very, very important topic that you should really understand and really know, because the past paper questions on these topics, it's so frequent you can almost predict it's going to come every year. So yeah, look out for these. So polysaccharides are large carbohydrates formed from many monosaccharide units joined by glycosidic bonds through condensation reactions. So the starch and cellulose are both polymers of glucose. However, they are different. So how do they differ between each other? It's the type of glucose, the glycosidic bonds involved, the 3D structure of the starch and the cellulose, and the biological function they perform. Starch is a storage polysaccharide found in plants, and it is made of alpha glucose monomers only. So they exist as a mixture of two polysaccharides. Again, you guys should really know this. So they are made of amylose and amylopectin. So amylose are unbranched, which means they are one long linear chain with their side branches, and they are alpha glucose joined by 1 to 4 glycosidic bonds. So with amylose, what happens is they coil into helical structure. So now to look at amylopectin, they are branch structures, which means they have side chains, and they consist of two types of bonds. So they have alpha 1 to 4 glycosidic bonds, and you have 1 to 6 glycosidic bonds at the branch points. What is the function of starch? So they are compact and insoluble, so this makes them suitable for storage. Again, you guys should really know this slide as well. I'm emphasizing it because it's just so frequent. You will see a question on this almost every single year. So the helical amylose allows tight packing, and the branched amylopectin provides many ends for rapid enzyme action. So this is what makes starch such a good form of storage in plants. So cellulose is a structured polysaccharide in plant cell walls. So remember, so starch is more to do with the storage side of plants, and cellulose is more on the plant cell structure side. So that's how they differ in function. But anyway, back to cellulose. So it's a structured polysaccharide in plant cell walls, and they're made of beta-glucose monomers. So beta-glucose molecules are joined by beta 1 to 4 glycosidic bonds. And remember to distinguish between what glycosidic bonds are if you're asked to differ between starch and cellulose. Starch always has alpha 1 to 4 glycosidic bonds, or alpha 1 to 6 glycosidic bonds, depending on what you're talking about, amylose or amylopectin. Cellulose, they always have beta 1 to 4 glycosidic bonds. So every alternate beta glucose is rotated 180 degrees. So if you look at the monomer level of cellulose, the first monomer will be upright, and the second one is going to be rotated 180 degrees. And then the third monomer will be back to the original form, and then the fourth one will be again rotated 180 degrees. So they alternate in structure basically, and this results in long straight unbranched chains. And microfibrils. So each beta glucose molecule has OH groups. Just before we go back into this, it's quite important for you guys to know how to draw alpha glucose monomers, beta glucose monomers. You should be able to draw the polypeptide chains of the alpha glucose monomers, and the beta glucose monomers as well. Another thing is you need to be able to show the condensation reaction happening between the different monomers as well. So do look at your textbooks and regularly practice basically practice drawing them out. So anyway, going back to this, so hydrogen bonds form between OH groups of adjacent cellulose chains, and many parallel cellulose chains are held together by hydrogen bonds to form cellulose microfibrils. So the properties of microfibrils are they are extremely strong, they have high tensile strength, and they are resistant to stretching. Function of cellulose. So this provides mechanical strength to plant cell walls, and cellulose prevents cell bursting during osmosis, and this allows cells to remain turgid, and it supports the overall plant structure. That's a SNAB today. Hope you found this video useful. If you have any questions or if you need further clarifications, let me know in the comments below and I'll get back to them as soon as possible. And don't forget to like, share, and subscribe.